A cell's ploidy indicates the number of each type of chromosome present. Think of ploidy in terms of shoes. Organisms whose cells contain just one of each type of chromosome are called haploid. In other words, you only have one of each shoe, either a right shoe or a left shoe. Those whose cells contain two versions of each type of chromosome are termed diploid, pairs of shoes in this case. Diploid cells have one paternal chromosome and one maternal chromosome. Organisms with three or more versions of each type of chromosome are called polyploidal. This could be 3N, 4N. Most of the time, at least with animals, diploid is about all you're going to get. Although we do see triploid, quadruploid, and octoploid in certain plant species. In animal and plant zygotes, the maternal and paternal sets of chromosomes line up to make the zygote's new set of DNA. So mitosis, as discussed previously, is a form of asexual division of somatic cells in which the diploid mother cell produces two genetically identical diploid daughter cells. Meiosis is a form of sexual division of gametes which takes place in two steps. Meiosis I is the division of a diploid mother cell's nucleus into two haploid daughter cells. Meiosis II and cytokinesis is the second division of haploid cells into four separate gametes, either sperm or egg. You might be thinking, shouldn't the cell's DNA from the first division be split in half again? Technically, no. Division 1 goes from 2N to N, but there are actually two copies of the N's which get distributed during the second division. Confused? Think of meiosis as a two-page test where I have two copies of each page. In the beginning, though I have four separate pages, I have only two unique pages, in other words, 2N. So in humans, the egg or sperm mother cell has 23 pairs of chromosomes. After the first division, both copies of page 1 and both copies of page 2 separate. Now you have two pages separated, but each one is only a unique page. In humans, though there are two copies of a chromosome, there's only one unique chromosome amongst the pair. This would also be like a cell with two left shoes of the same type or two right shoes of the same type. After the second division, each individual page is now separated into their own areas. You have one page each now, but the unique number of pages is still one. This is why the haploid number of chromosomes does not change between meiosis I and meiosis II. In humans, not only are the gametes haploid, there's only one copy of the haploid chromosome. So let's go through the stages of meiosis and show how this contributes to genetic diversity. Before beginning, please note that interphase in mitosis and meiosis is nearly identical, so I don't need to talk about the interphase here. If you need to, please check my mitosis videos. The mother cell is a diploid sperm or egg cell in animals. A lot happens in Prophase I, which is why I split this up into two slides. In early Prophase I, chromatin converts to homologous pairs of chromosomes. The nucleus and nucleolus disintegrate while the centrosomes head towards the poles of the cell. These chromosomes then join together by synapsis. The structure that results from synapsis is called a tetrad, consisting of two homologues. The chromatids of the homologues are called non-sister chromatids. Maternal and paternal chromosomes are non-sister chromatids because they could differ slightly in the genes on them. For example, one chromosome may carry a gene for hitchhiker's thumb. The other chromosome pair may not, and that helps to determine whether or not you have hitchhiker's thumb. Later in Prophase I, these non-sister chromatids begin to separate. The exchange or crossing over between homologous non-sister chromatids occurs. After replication, sister chromatids stay tightly joined along their entire length. When homologues synapse, two pairs of non-sister chromatids are brought close together and are held there by a network of proteins called the synaptonemal complex. Crossing over occurs when chromosomal segments are swapped between adjacent homologues. At each point where crossing over occurs, the non-sister chromatids from each homologue get physically broken at the same point and attached to each other. As a result, segments of maternal and paternal chromosomes are swapped. Know how letters A, B, and C represent different genes on the chromosome. Crossing over allows for alleles or alternate versions of the same gene to be swapped, making the chromosomes more diverse. Crossing over can occur at many locations along the length of each synapse homologue. Both sets of non-sister chromatids may undergo crossing over, resulting in the swapping of segments between maternal and paternal chromosomes. Crossing over produces new combinations of alleles on the same chromosome, combinations that did not exist in each parent possibly. It also forms genetic recombination that helps to increase genetic variability of gametes produced by meiosis beyond that produced by random assortment of chromosomes. So after Prophase I, we head into Metaphase I, and the crossed over tetrads line up at the metaphase plate. In this stage, another important mechanism occurs that increases genetic diversity: independent assortment. Independent assortment was first coined by geneticist Gregor Mendel, who stated that genes are inherited independently from one another. We now know that since genes are housed on chromosomes, the way in which they line up has an effect on the gametes made. For example, this set of chromosomes could line up this way and give us this set of gametes. It's quite possible that those same chromosome pairs lined up this way, giving us a genetically distinctive set of chromosomes from the first scenario. In fact, one person can produce 8.4 million genetically distinct gametes from independent assortment alone. This comes from the fact that there are 2 copies of each chromosome, and we have 23 pairs of them, so the number comes from 2 to the 23rd power, or approximately 8.4 million gamete combinations. This does not account for crossing over events from Prophase I. You'll notice that the rest of meiosis tends to mimic the mechanics of mitosis. So in anaphase I, the paired homologues separate and begin to migrate to opposite ends of the cell. In telophase I, the homologues finish migrating to the poles of the cell. Then the cell divides in the process of interkinesis, since this is the split between meiosis I and meiosis II. So both stages happen immediately after one another. The end result of meiosis I is that one chromosome of each homologous pair is distributed to a different daughter cell. Thus, a reduction division has occurred. The daughter cells of meiosis I are haploid and are still in the form of sister chromatids. Now we are ready for meiosis II, which starts with Prophase II, in which the spindle apparatus forms and one spindle fiber attaches to the centromere of each sister chromatid. Notice here that the cells will have similar mechanics to mitosis at this point, as the goal of meiosis II is to divide the sister chromatids into individual chromosomes. In metaphase II, the replicated chromosomes line up at the metaphase plate. In anaphase II, sister chromatids separate. The resulting daughter chromosomes begin moving to opposite sides of the cell. In telophase II, chromosomes arrive at opposite sides of the cell. A nuclear envelope forms around each haploid set of chromosomes, and each cell gets ready to undergo cytokinesis. So take a look. None of those potential daughter cells will have the exact same DNA. And as those chromosomes relax, they go back to being chromatin, and then we're going to get ready to completely divide by cytokinesis. So in meiosis II, results in 4 haploid cells, each with one of each type of chromosome. Thus, one diploid mother cell with replicated chromosomes gave rise to 4 haploid daughter cells with unreplicated chromosomes, and these eventually become the sperm and egg cells used in animals and plants to help fertilize and make zygotes. Genetic variation in sexual reproduction results from independent assortment and crossing over. Separation and distribution of homologous chromosomes during meiosis I can result in a variety of combinations of maternal and paternal chromosomes. Each daughter cell gets a random assortment of maternal and paternal chromosomes, and thus genes, which generate a great deal of genetic diversity in the subsequent gametes. Fertilization allows for even more genetic diversity, as it combines the effects of meiosis in 2 parents, making an offspring that is genetically distinct from each parent. How distinct? In a single human with a haploid chromosome number of 23, you can produce approximately 8.4 million different combinations of chromosomes in your gametes from independent assortment. Combine that with an 8.4 million different combinations from a second parent, and you end up with 70.6 million genetically distinct zygotes that can be produced from two parents. This does not take crossing over into consideration, so the zygote combinations would be limitless. What I am trying to say is, unless you are an identical twin, you are truly unique. No one on this planet has ever been like you, nor will there ever be anybody like you on this planet. Even if you are an organism like a plant that can self-fertilize, the genetic variation introduced during meiosis ensures that even where gametes from the same individual combine, the offspring will be genetically different from the parent. Up next, we will see what happens when meiosis undergoes errors and the chromosomal disorders that follow.